My name is Rezvani, and I've done work in this past summer on the directed evolution of novel G-protein coupled receptors for orthogonal binding with select ligands. And in order to demystify what that means, you first have to know what G-protein coupled receptors are. So GPCRs are a membrane receptor, which means that they're a protein that exists on the membrane of the cell, and they will bind selectively to ligands, which are usually small molecules. And upon activation by a ligand, a G-protein coupled receptor interacts with a G-protein, you might expect, a couple, and sets off a pathway. And these pathways are found in all sorts of eukaryotic organisms, and they control a lot of neurochemical activity, cell-cell interaction, regulatory mechanisms in organisms, and a lot of previous neuropharmaceutical research has been done in designing or binding ligands that either activate or inhibit G-protein coupled receptor pathways. And you can see the structures of two of them right here that overlaid, the muscarinic receptors 2 and 3. This is in a big paper by Brian Kukulka, who actually got a Nobel Prize for finding crystal structures of GPCRs. And that yellow and red blob thing is acetylcholine, and it's the ligand of the muscarinic receptors. So it goes inside and interacts with the helices. So basically, GPCRs, because they're a protein, there's a corresponding gene that you sequence, you can modify, and thus downstream you would be modifying the amino acid sequence of the GPCR. And this diagram right in the top right is a overhead view of the 7 transmembrane domains. Yeah, this right here is the these are the 7 transmembrane domains of the GPCR, and in green is the ligand binding pocket, which exists somewhere in the mid-level of the helices. And essentially, by changing the identity of some select amino acid residues, you change hydrophobicity, you change charge, and most importantly, you change the resulting ligand binding pocket, which means you can evolve a receptor to bind to ligands of your choice, which is kind of like a lock fitting the key. The opposite direction, opposite approach taken, where would be to just try to find ligands that bind at an existing receptor. And an interesting application of this in the bottom right here is the molecule DNT, and one of the receptors I'm trying to work with, MRR226. Naturally responds to this molecule, and you can imagine that if you evolved the receptor, you could, and change its sequence, you can get it to respond to a close chemical relative, TNT, or the explosive that everyone's familiar with, and then you would be able to take this MOR226 modified version, and then using gene therapy techniques, you can imagine you can introduce it into an organism like Drosophila and basically create flies that could sense out TNT, and that's like some sort of like security application. But that's more of a fun sort of thing, whereas most applications would be for like pharmaceuticals. But yeah, so in order to understand why this is worthwhile, you have to understand what directed evolution is. So the idea is, like I said, you make modifications to the genetic sequence, and then that translates into changes and modifications in your protein library. And by applying a Darwinian selection of some sort, you basically choose the ones that work the best in your selection. And I'll get to what our selection is. But you choose the ones that work the best, and it's an iterative process where those become the template for the next round. And the idea is to basically emulate natural selection, but just sped up. so it'll take millions of years. And I use yeast for this because I work with mammalian receptors, but mammalian cells are not an ideal chassis for this kind of work because they're slow and difficult to use. So right here, you see a simple yeast GPCR pathway. In fact, the only major one that yeast has. And previous work in the past has been done to strain engineer the yeast to accept this. H stands for human. So basically, mammalian GPCRs can now be expressed in this yeast. And most importantly, the function of the GPCR is correlated to the yeast survival. This gene right here, HIS3, is an oxytrophic marker, which means that it needs to be activated in order for the yeast to survive, provided you have the correct conditions. So by linking the survival of the yeast to the activity of the GPCR pathway, you create a system by which you can actually select for GPCR activity, and you can do directed evolution. So work has been done in the past, like I said, and a lot of what I've been doing is just to set up this system, use the strain of yeast, get it to work, introduce different GPCRs into it, and make sure they're functional. So in the blue is the wild type receptor, the muscarinic receptor 3 in the yeast, and its native ligand is acetylcholine. And as you can see, OD650 is just a measure of basically culture density, so it's how growth. And the yeast have grown, and they do not respond to, or the dread yeast do not respond, and dread refers to the mutant yeast that basically have 2 key amino acid changes in the sequence that affect the ligand binding domain. And CNO is a different ligand that does activate. It's the ligand they were evolving towards, and you can see that response that dread has that the wild type doesn't have. So that's good. And this is actually important because you have to be able to run your selection screens on solid phase media. So this is just a similar validation for the wild type that you get colonies on acetylcholine, and that's cool. So one way that I would want to basically expand the power of directed evolution, and I'm trying to do so right now, is to use better library designs. In the past, error-prone PCR has been used, and essentially that's a way of amplifying a DNA sequence and introducing random point mutations along the way. But it's non-specific, and you don't have much control over what you're mutating, so you imagine you get a lot of nonsense mutations that will either be deleterious to structure, deleterious to trafficking to the membrane, and you're not going to be able to affect what you necessarily want, which is how it binds to ligands. So I've been working on creating other libraries, and I'll describe some of these now. So cassette mutagenesis, and the idea here is that if you focus on the areas in the genetic sequence of the GPCR that are known or thought to interact with ligands, you can introduce variations there, and then you develop a more focused library to work with that you can then screen using that selection mechanism described earlier. And I synthesized gene fragments, ligated them together, and then you get this, in the red is just like where the variations would occur. And essentially the red is, you can think of it as like an amino acid degeneracy. So assume that at one point in time, it was, the wild type sequence, there was an alanine, but now I've allowed for there to be any of the 20 major amino acids, including the synthetic ones, but yeah. So additional strategies that I plan on implementing in the future. Right here is just a visual description of a technique called homologous recombination, where you mix and match different features from a similar set of proteins, and you get this novel thing that has variations, but should not compromise structure, since all of these proteins are related enough that there's a certain degree of structural integrity between them. And here is a picture of a linear plasmid system that I hope to utilize that's really, really new, that basically would allow for in vivo continuous evolution that would also be desirable. So as far as future works, I'm in the process of doing evolution and selection rounds, and as well as troubleshooting. I'm looking around for additional candidate receptors. Right now I'm also trying to get olfactory receptors, which are a kind of GPCR, to express properly, and I'm also considering chemokine receptors. And then when ideal receptors are evolved, you need to characterize them further, so that would be done in mammalian cell lines like X cells, and then you can also do electrophysiology experiments with frog eggs. And yeah, there's a list of people to acknowledge. Yeah. So yeah, it's done.